Hello, everyone. Well, I learn something new every day by reading Mel's post. So I thought I would make a video to talk about what I just learned. Um, okay, you thought they were really offering you protection, and they just keep forgetting to tell you they knew they gave you all the real cause for decades. And this post was from 6 years ago. Your so called antibodies, are much more than they cracked them up to be. And negative does not negate infected. So this link is to Prion-like domains and RNA binding proteins are essential for building subnuclear paraspeckles. Okay. Prion-like domains, or PLDs, are low complexity sequences found in RNA binding proteins associated with the neurodegenerative disorder amyotrophic lateral sclerosis. Recently, PLDs have been implicated in mediating gene regulation via liquid phase transitions that drive ribonucleoprotein granule assembly. In this paper, we reported many PLDs in proteins associated with paraspeckles, subnuclear bodies that form around long non-coding RNA. We mapped the interactome network of paraspeckle proteins, finding enrichment of PLDs. We show that one protein, the RBM14, connects key paraspeckle subcomplexes via interactions mediated by its prion-like domains. We further showed that the RBM14 prion-like domain, as well as the PLD of another essential paraspeckle protein, the FUS, is required to rescue paraspeckle formation in cells in which their endogenous counterpart has been knocked down. Similar to FUS, the RBM14 prion-like domain also forms hydrogels with amyloid-like properties. These results suggest a role for prion-like domain mediated liquid phase transitions in paraspeckle formation, highlighting this nuclear body as an excellent model system for understanding the perturbation of such processes in neurodegeneration. Low complexity regions containing biased amino acid composition are abundant in the proteome, particularly in regulatory molecules, yet the utility and molecular mechanism of these largely unstructured regions have been elusive. Prion-like domains are a subset of low complexity regions enriched in uncharged polar amino acids and glycines, with similarities to the yeast prion protein that can be defined using a hidden Markov algorithm. Prion-like domains are often found in RNA binding proteins that drive protein aggregation in neurodegenerative disorders such as amyotrophic lateral sclerosis, ALS. To fully appreciate their pathophysiology and guide therapeutic strategies, we need to understand the normal physiological role of PLDs. An emerging concept is that PLDs allow proteins to functionally aggregate, forming higher order assemblies and microscopically visible RNP granules. Concentrating proteins and RNA in a constrained space is believed to result in more efficient gene regulatory processes. The biophysical properties of prion-like domain mediated interactions may explain liquid-like properties of RNP granules, as PLDs expressed in vitro form mesh-like networks manifesting as hydrogels. These hydrogels are reminiscent of, but different to, amyloid material and may represent a functional amyloid. Central to these discoveries has been research into the RNA binding protein FUS fused in sarcoma with a prion-like domain containing many repeats. The central tyrosine in the FUS repeats is essential for hydrogel formation and stress granule targeting in the cytoplasm. Like cytoplasmic RNP granules, nuclear bodies also assemble without a membrane and are composed of proteins and RNA molecules selectively recruited based on protein-protein and protein-RNA interactions, although the molecular basis of these interactions is unclear. Paraspeckles are a nuclear body that are seated by and built on the long coding RNA neat, nuclear paraspeckle assembly transcript. There are 40 known paraspeckle proteins, most abundantly nuclear RNA binding proteins enriched in RNA recognition motifs, RRMs, zinc fingers, K homology domains. Paraspeckle formation is triggered by transcription of neat, followed by the recruitment of different proteins that coordinatingly built up the paraspeckle structure. Intriguingly, FUS, TDP43, SS18L1, HNRNPA1, TAF15, and EWSR1 are all genes encoding paraspeckle proteins that contain prion-like domains and have known ALS-causing mutations. Paraspeckles are also apparent in ALS motor neurons, and NEAT1 is upregulated in the related condition frontotemporal lobar degeneration. Furthermore, paraspeckles are stress-responsive structures induced by viral infection, proteasome inhibition, and differentiation. Paraspeckles influence gene expression by nuclear retention of RNA with inverted repeats and by sequestration of specific transcription factors. Given many RNA binding proteins with prion-like domains contain nuclear localization signals, we speculated that the prion-like domains play a role in nuclear body formation. To identify protein-protein interactions likely mediated by the prion-like domains, we mapped the interactome network of paraspeckle proteins. We found that RBM14, RNA binding protein 14, is an essential paraspeckle component, mediates a key interaction linking several other essential proteins into the network. This interaction is driven via the PLD of the RNA binding protein 14, and we showed that this region possesses the same liquid phase transition potential as fused in sarcoma, forming hydrogels with amyloid-like properties in vitro. We also confirmed that the PLDs of both RBM14 and FUS are responsible for targeting these proteins to paraspeckles and are essential to the formation of paraspeckles. The paraspeckle protein-protein interactome is rich in prion-like domain containing proteins. To identify the direct protein-protein interactions underpinning paraspeckles, we performed a comprehensive yeast 2-hybrid screen on the known and some putative paraspeckle proteins. MAT-A yeast containing each candidate fused to the Alex-A DNA binding domain were mated with MAT-A yeast containing DNA activation domain fusions. The LACZ reporter was used to screen for interactions, and interaction probability scores were assigned in higher numbers, reflecting the most stringent interactions in numerous biological replications. Interactions between key proteins were validated by co-transformation of both plasmids, and in some cases by co-immunoprecipitation in HeLa cells. 53 interactions between 29 paraspeckle proteins were identified that met the most stringent scoring criteria for reproducibility and strength, and these are shown in a network at figure 1E. Paraspeckle proteins are usually enriched in prion-like domains as defined by the Prion-like amino acid composition tool. There's an overpresentation of proteins with prion-like domains in the interactome network. 66% of network proteins have prion-like domains, 19 out of 29, whereas 55 of the starting proteins had prion-like domains, 26 out of 47. I will put the link down below to this full article so you can check it out for yourself. We had previously performed short interfering RNA knockdowns of the majority of the paraspeckle proteins to determine their importance to paraspeckle formation, summarized in Table S1. Proteins that completely or partially ablate paraspeckles when knocked down are colored on Figure 1E and comprise over half the network. 52% of the network proteins are required for paraspeckle formation, whereas only 36 of the starting protein pool are in this category. illustrating the potential importance of protein-protein interactions in paraspeckle formation and maintenance. Furthermore, 12 of the 15 proteins required for paraspeckle formation that are in the interactome network also contain prion-like domains, indicating that these domains may play a key role. Prion-like domains are emerging as important modules in gene regulation that act through a process of functional aggregation and phase transition, thus explaining their presence in numerous RNA binding proteins, transcriptional regulators, and their fusions to DNA binding domains in chromosomal translocations in cancer. To investigate a potential for prion-like domain-mediated functional aggregation in the context of paraspeckle formation, we focused on the prion-like domain containing protein RBM14, identified as directly connecting two other essential proteins, the NO or the non-POU domain containing octamer binding protein, and the SFPQ splicing factor proline glutamine rich to the rest of the network. So the rest of this kind of gets technical, so I won't read this, but I do. They're using yeast, and the fact that these form hydrogels with amyloid-like properties in vitro. It's rather scary to me, but again, like I said, I want to point out. I see this name over and over in this, and no, it's not jumping out to me. The HeLa cells. Okay, let me read a little bit more. Nuclear body and RNP granule formation are highly dynamic processes, but the molecular basis for this remains unclear. Given the abundance of tyrosine and serine residues in prion-like domains, phosphorylation could play a role in regulating the prion-like domain-mediated interactions underpinning bodies. Suggestive of this, we observed different SDS page mobility for RBM14 prion-like domain mutants expressed in HeLa cells, and found evidence that the RBM14 PLD is phosphorylated in cells. Furthermore, the FUS PLD can be phosphorylated, and phospho-FUS hydrogels have weaker interactions. Beyond paraspeckle formation, the RBM14 is important in co-regulation of transcription and splicing, centriole formation, and DNA repair. Re-examining these papers, we find that the RBM14 prion-like domain required for 3 out of 4 of these functions, although the role of the RBM14 prion-like domain in DNA repair is yet to be assessed. It is an attractive hypothesis that functional aggregation is involved in the dynamic formation of localized DNA repair assemblies. Notably, another essential paraspeckle protein, the SFPQ, also implicated in DNA repair, requires functional aggregation via coiled coil motif for optimal DNA binding. The regulation of RNP granule formation is emerging as a potential therapeutic application for cancer and neurodegenerative disorders. Of particular relevance is ALS, associated with cytoplasmic aggregates of normally nuclear prion-like domain containing proteins, and caused in many cases by mutations within prion-like domain containing proteins. Also important is the recent discovery of the most common genetic cause of ALS, a repeat expansion in C9orf72, resulting in formation of toxic nuclear RNA granules that sequester RNA binding proteins and production of mutant proteins that bind hydrogels. Paraspeckles now join stress granules and C9orf72 foci as RNP granules implicated in ALS pathobiology. 6 paraspeckle proteins are known to cause ALS when mutated. It is thus possible that other paraspeckle proteins are putative candidates for novel ALS genes. This growing appreciation of the importance of functional aggregation mediated by prion-like domains in RNA binding proteins in ALS is opening the door for future studies into how these processes are perturbed in disease. Until now, the common feature for paraspeckle proteins was the presence of RNA binding domains, whereas the long non-coding RNA NEAT1 was thought to be the key structural determinant of paraspeckle formation. Our data do not diminish the importance of the NEAT1, rather they suggest that in addition to protein-RNA interactions, there are also prion-like domain-mediated protein-protein interactions, and together, these give rise to a dynamically regulated structure. Indeed, many paraspeckle proteins associated within cells in RNA-dependent manner. Questions remain about the repeat motifs within different prion-like domains and how differences in these relate to functional aggregation into RNPs and other assemblies. Paraspeckle targeting could join yeast aggregation and toxicity as a useful assay to address the relative contributions of different residues within PLDs to functional aggregation. So, their methods and materials: two hybrid yeast, two hybrid interaction screen, and of course, the HeLa cells. Bacterial plasmids. HeLa cells were grown in fetal calf serum, penicillin, and streptomycin. They were transfected with plasmids using lipofectamine 2000 from Life Technologies, according to the manufacturer's instructions. For imaging experiment, cells transfected with plasmids followed by fixation the next day. For RBM14 short interfering RNA knockdown experiments, cells were grown on cover slips, 8 well plates, and then transfected with the short interfering RNA against RBM14 using lipofectamine, RNA interference, or RNA IMAX, according to the manufacturer's instructions. So if you watch some of my earlier videos, I talked about the short interfering RNAs and RNA interference and this RNA IMAX. The rain, and I talked about how when I see this RNA interference, I can't help but see the rain. The rain is coming, and one of the monoclonal antibodies that they were using, Regeneron is using, was literally REGN, another rain. I can't help but think of the days of Noah and the flood that was coming. There was a rain coming, and he tried to warn people, and they didn't believe him, and they didn't listen. So, your so-called antibodies are much more than they crack them up to be, and negative does not negate infected. So let's see. She also put a couple more comments on here. So, Trojan horses all fall down. Let's see what that is. Excuse me. Well, she talks about extracellular vesicles as modulators of cell-to-cell communication in healthy and diseased brain. Homeostasis relies heavily on effective cell-to-cell communication in the central nervous system, probably more so than in other organs. Such communication is crucial to support and protect neurons, especially during aging, as well as to control inflammation, remove debris, and infectious agents. Emerging evidence indicates the extracellular vesicles, including endosome-derived exosomes and fragments of the cellular plasma membrane, play a key role in intercellular communication by transporting messenger RNA, microRNA, and proteins. In neurodegenerative diseases, secreted vesicles not only remove misfolded proteins, but also transfer aggregated proteins and prions, and are thus thought to perpetuate diseases by infecting neighboring cells with these pathogenic proteins. Conversely, in other CNS disorders, signals from stress cells may help control inflammation and inhibit degeneration. EVs may also reflect the status of the central nervous system and are present in the cerebrospinal fluid, indicating that exosomes may act as biomarkers of disease. That extracellular RNA, and in particular microRNA, can be transferred by EV also indicates that these vesicles could be used as carriers to specifically target the central nervous system to deliver immune modulatory drugs, neuroprotective agents, and anti-cancer drugs. Here we discuss the recent evidence indicating the potential role of exosomes in neurological disorders and how knowledge of their biology may enable a Trojan horse approach to deliver drugs into the central nervous system and treat neurodegenerative and other disorders of the central nervous system. Adding more infectious junk DNA to the mix is not the answer, she says. So it's highly unique to watch so many people over the years describe spirochetal disease in so many different ways. And let's open this link. Okay, proteomics and analysis of amyloid and non-amyloid prion disease phenotypes reveals both common and divergent mechanisms of neuropathogenesis. Prion diseases are a heterogeneous group of neurodegenerative disorders affecting various mammals, including humans. Prion diseases are characterized by misfolding of the host-encoded prion protein into a pathological isoform termed PRPSC. In wild-type mice, prion protein is attached to the plasma membrane by a GPI anchor, and the PRPSC typically accumulates in diffuse non-amyloid deposits with gray matter spongiosis. By contrast, when mice lacking the GPI anchor are infected with the same prion inoculum, the prion protein or the PRPSC accumulates in dense perivascular amyloid plaques with little or no gray matter spongiosis. In order to evaluate whether different host biochemical pathways were implicated in these two phenotypically distinct prion disease models, we utilized a proteomics approach. In both models, infected mice displayed evidence of a neuroinflammatory response and complement activation. Proteins involved in cell death and calcium homeostasis were also identified in both phenotypes. However, mitochondrial pathways of apoptosis were implicated only in the non-amyloid form, while metal binding and synaptic vesicle transport were more disrupted in the amyloid phenotype. Thus, following the infection with a single prion strain, prion protein anchoring to the plasma membrane correlated not only with the type of prion disease or prion misfolding deposition, but also with the unique biochemical pathways associated with pathogenesis. Are you paying attention to real science? And of course, we're going to go extremely slow. There we go. Gamma herpes infection of human neuronal cells. Eusyndrome, psych, cancers, and addiction. People paying attention? So let's look at the gamma herpes infection of neuronal cells. Gamma herpes viruses, human herpes virus 4, HHV4, and HHV8 are two prominent members of the herpes virus family associated with a number of human cancers. HHV4, also known as Epstein-Barr virus, a ubiquitous gamma herpes virus prevalent in 90 to 95% of the human population, is clinically associated with various neurological disease such as primary central nervous system, lymphoma, multiple sclerosis, Alzheimer's disease, cerebellar ataxia, and encephalitis. However, the possibility that Epstein-Barr virus and Kaposi sarcoma-associated herpes virus, KSHV, can directly infect neurons has been largely overlooked. This study has, for the first time, characterized Epstein-Barr virus infection in neuronal cell backgrounds by using the SHSY5Y neuroblastoma cell line, teratocarcinoma, entera neurons, and primary human fetal neurons. Furthermore, we also demonstrated that the KSHV infection of neural SY5Y cells, these neuronal cells, were infected with green fluorescent protein-expressing recombinant Epstein-Barr or Kaposi sarcoma. Microscopy, genetic analysis, and immunofluorescence and Western blot analysis for specific viral antigens supported and validated the infection of these cells by EBV or KSHV and showed that the infection was efficient and productive. Progeny virus produced from infected neuronal cells efficiently infected fresh neuronal cells as well as peripheral blood mononuclear cells. Furthermore, acyclovir was effective at inhibiting the production of virus from neuronal cells, similar to lymphoblastoid cell lines. This suggests active lytic replication in infected neurons in vitro. These studies represent a potentially new in vitro model of Epstein-Barr virus and KSHV-associated neuronal disease development and pathogenesis. So, when we go over here, she says, there's no such thing as viruses. They are lytic. We just read that. Bacterial associated stress-induced morphological forms of infinite antigen variations in less than 2 seconds under antibiotic stress. I'll put the link down below to this video. I opened it a while ago and it just threw my computer off, so I'm not even going to try to do that again. The stealth, the U.S. Army called a bioweapon in 1965, they gave to everything. So let's go here. Sorry. Okay. This page has so much information. The lipopolysaccharide-induced conversion of recombinant prion protein. The real cause of AIDS, immune suppression. They gave us in vaccines. Because you're already infected with immune-suppressing cells of Epstein borreliosis and so-called normal prion protein. Henrietta Lacks. Immortal HeLa cells, the turd in the punch bowl, used for all vaccines for decades, had syphilis, and they knew spirochetal prion proteins could not be killed, as testified before Congress. Coexistence of ribbon and helical fibrils originating from human island amyloid HIA-PP 20 through 29 revealed quantitative nanomechanical atomic force microscopy, uncontrolled misfolding of proteins leading to the formation of amyloid deposits, and it's associated with more than 40 types of diseases, such as neurodegenerative diseases and type 2 diabetes. All right, let me get down. Here we go. The U.S. Army, they knew all spirochetal AIDS. Okay, so I'm going to open up this whole link. It's going to go especially slow. Okay. Borrelia, human relapsing fever, and parasite vector host relationships. And remember, this is from 1965. Borrelia, pathogenic to man, are the causative agents of relapsing fever and its varieties known as tick fever, Garib-Gaz, Karopata, Kimputu, Gorgonia, Billus typhoid, fowl nest fever, and vagabond fever. It may be divided into louse-borne, which is usually epidemic, and tick-borne, which is mostly endemic. Exceptions occur. Human Borrelia infections are or were seen all over the world except in Australia, New Zealand, and Oceania. And I think I read this before in one of my videos because I commented how they probably fixed that by now. That's probably not true anymore. So tick-borne Borrelia infections remain mostly restricted to the vicinity in which this one, the ornith, I don't even know how to pronounce that, lives. And again, does not spread further than the infected. That's a lie. These ticks have spread everywhere. Don't let them tell you that, oh, there's no tick-borne diseases here. So all of this, she cycles through these posts and has been for years. And there's just so much information, but I can still learn something every day by following her. I will put all the links down below. And again, thank you for watching.